class. Today I will start class 12 chapter. The chapter name is chapter Molecular Base of Inheritance in Genetics and Evolution, class 12. Okay, this is the largest chapter, but you remind that students at a bhabiswa. Yet the classification kora hoise class it is a chapter number 6 hoy. Kintu from exam point of view, exam or follow up nitish or about malke examiners to chapter that important chapter. Yet pray 35 to 45 to month period like around schooling or class code. Or pray 20 marks month more schooling or it's out of 45, 25, 20 marks to 25 marks is a prior. But 50% গোটেই তোমালোকৰ biology course এই chapter cover কৰিছে genetics and evolution. In genetics you will find two chapters. Genetics তোমালোকে দুটা chapter পাবা. Chapter number 1 is principles of inheritance and variation. Principles of inheritance and variation. Principles of inheritance and variation. This is chapter number 1 in genetics. And the second chapter is molecular basis of inheritance. Molecular basis of inheritance. Basis of inheritance. Molecular basis of inheritance. Okay, it is one of the most interesting and most important chapter from exam point of view. So today I will touch this chapter, molecular basis of inheritance, genetics and evolution, the largest chapter from class 12 biology, for class 12 biology. Molecular base of inheritance. Okay. What this chapter will deal with? This chapter keep what this chapter will deal with? Molecular base of inheritance. In this chapter, you will learn about the structure of genetic material and the experimental proofs that are carried out, experiments that are conducted in order to find a proof that DNA is genetic material, or RNA to some extent also genetic material in certain organisms. So then thereafter we study about the chemical structure of genetic material. Actually, the first stress should be given that DNA is genetic material and structure of the DNA molecule, structure of RNA. But you remind one thing that RNA is genetic material for only very few numbers of organisms, mainly viruses. That genetic material সাধাৰণতে genetic substance, it holds DNA. কিন্তু সকলোবিলাক organismত DNA genetic material নহ'ব পাৰে. For example, certain RNA viruses are there where genetic material is RNA. Example of RNA viruses are XIV, human immunodeficiency virus. Its genetic material is RNA. And similarly, RSV, Ross sarcoma virus, its genetic material is RNA. So TMV, tobacco mosaic virus, its genetic material is RNA, and so on and so forth. These are some examples of RNA containing viruses. Like RNA thoka virus hoy. Kintu maximum virus sadhanতে আমি bacteriophage নামৰ এটা গ্ৰুপত ক্লাসিফাই কৰো. এই গ্ৰুপটো bacteriophage কোৱা হয়. Bacteriophage গ্ৰুপত কিন্তু আটাইখিনি genetic material DNA থাকে. Means bacteriophages are bacteriophages. Here genetic material is genetic substance is DNA. Whereas HIV, RSV, tobacco mosaic virus, these are genetic material is RNA. RNA acts as genetic material. RNA is genetic material in this group. So what we are getting that genetic material can be DNA, can be another, preferably the genetic substance in almost all organisms are DNA. So starting from bacteria onward, from bacteria up to human being, the genetic substance is DNA. From bacteria up to human being, genetic substance is DNA. It means bacteria means among the prokaryotes. Thereafter protista, fungi, plant, animal, whatever kingdom we consider genetic material is DNA. So my objective of this chapter, basic objective of this chapter is to deal with the structure of DNA molecule. First, structure of DNA molecule. Let us speak about structure of DNA. Thereafter, I will deal with the structure of RNA, and thereafter experiments carried out by different scientists to find out the genetic substance is universally DNA. Later on I will discuss about that. So first is structure of DNA. Structure of DNA. Also later on, also of RNA. I will deal later. Okay. So DNA, as a name implies, perhaps DNA and RNA, both are we know as nucleic acids. Both of these two are called nucleic acids. These two are nucleic acids. This what similarity between DNA and RNA is that they are nucleic acids. Nucleic acids were first of all discovered. Nucleic acids first discovered by Frederick Miescher in 1869. Okay. In 1869, it was Frederick Miescher who first of all isolated a specialized group of biomolecules from the nuclei of past cells. He isolated a specific group of biomolecules from the nuclei of past cells. Okay, these substances, since they are extracted from nuclei of past cells. Now, do you know what is exactly past cell means? Past cells, they are nuclei of WBC nuclei. Actually, past cells are worn out WBC. They are worn out WBC from whose nuclei Frederick Miescher isolated nucleic acids. Thus, being the substances isolated from nuclei of WBC or past cells, the term initially to these substances are given not nucleic acid but as nuclein. So, nucleic acids were discovered by Frederick Miescher from the nuclei of past cells in 1869, but he coined the term nuclein rather than nucleic acids. Remember this. What I have spoken, again repeating, that nucleic acids were first of all discovered, actually extracted and hence discovered by Frederick Miescher in 1869, being isolated from the nuclei of worn out WBC, which we call as past cells. Since they are obtained from nuclei of past cells, they are named as nuclein rather than nucleic acids. Nuclein called. So nuclein was the nucleic acids ultimately. This is specialized groups of biomolecule not discovered so far so long up to 1869. 1869 discovered one molecule, a group of molecules that called nuclein. Frederick Miescher discovered person. Who will be given the credit for discovery of nucleic acids? Frederick Miescher, year 1869. Information number 1. Frederick Miescher, 1869, isolated nucleic acids, but he has given the term nuclein rather than nucleic acids. But later on, it was Altman after Frederick Miescher. After Frederick Miescher, it was Altman in 1889. Altman studied detailed the chemical properties of nuclein and found that nuclein is acidic in nature. Nuclein acidic in nature observed by Altman. And therefore immediately the nuclein term is converted into nucleic acids. That means the name nuclein is given by Altman as nucleic acids. So discovery of nucleic acids credited to Frederick Miescher, but the term nucleic acid was given by Altman. 2 points you remind. এটা হল নিউক্লিক এসিডস নিউক্লিন হিসাবে ডিসকভাৰ কৰিছিল ফ্ৰেডাৰিক মিescher, কিন্তু টাৰ্ম নিউক্লিক এসিড অৱশ্যে অল্টমানেহে দিছিলে after repeated study and found out that the acidic molecules. It more like information number 1. I think you might be obtaining therefore. Altman nucleic acids and Frederick Misser nuclein. Thereafter, contribution number 1 is by Frederick Misser. Contribution number 2 on nucleic acid discovery was Altman. Then in 1884, much before 1818 and much before the discovery of nucleic acid nature, it was Hartwig in 1884 observed that the substance which are responsible for controlling the parental characters in subsequent generations are present inside the nucleus. The concept that genetic material present inside the nucleus responsible for controlling the parental traits from one generation to successive generation. That concept of transmission of genetic character from generation to generation associated with this nucleic acids or nuclein was first of all given by Hartwig. But this contribution made by Hartwig, his credit can be given to Hartwig that he is the first worker to relate functional relationship with nucleic acids as a genetic substance. The genetic substance being nucleic acid first of all given, explained by Hartwig. That nucleic acids are genetic substance given by Hartwig. Okay, next thereafter, after Hartwig, chemical investigation of the structure of the DNA molecule was carried out. But later on, it was, however, Arthur Kornberg et al., sometimes called Kornberg et al. Kornberg was the first scientist who was able to synthesize DNA in vitro conditions. Kornberg et al. Was the first scientist capable of synthesizing DNA in vitro. In vitro should be in italics, it is shown. In vitro means in laboratory conditions. In laboratory conditions means without using template. Without using a template. Without using a template. Okay. মানে DNA নোহোৱাকৈ DNA synthesis process to প্ৰথমে start কৰিছিল Kornberg. And Kornberg was credited for discovering the Kornberg enzyme. The Kornberg enzyme is DNA polymerase. DNA polymerase. This is the enzyme responsible for synthesis of DNA in vitro conditions. Kornberg discovered or synthesized the DNA polymerase enzyme first. So DNA polymerase, in presence of DNA polymerase enzyme, that DNA can be synthesized without using a template, means without using a parent DNA molecule. With the help of DNA polymerase, you can synthesize DNA. That concept was given by Kornberg. And in vitro, so in laboratory condition, without using a living condition, you can synthesize DNA, was first of all shown by Kornberg. So Kornberg, another contribution made by. Then some other workers like Watson and Crick, 1953. Watson and Crick, along with Maurice Wilkins and Rosalind Franklin. Some other scientists that can be referred here because they're associated with discovering certain properties of DNA. Actually, the work made by Rosalind Franklin and Maurice Wilkins proved that the structure of the DNA molecule, actually their informations were used by Watson and Crick, and finally they proposed the double helical model of the DNA. Watson Crick is known for proposing the model of structure of the DNA molecule, which we call as double helical model of DNA. Double helical model of DNA. Double helix model of DNA. The structure of the DNA molecule, therefore, first of all, proposed by Watson and Crick. But raw material for their information, for their model, was given by Maurice Wilkins and Rosalind Franklin. Rosalind Franklin, along with Maurice Wilkins, were capable of taking an X-ray crystallographic picture of the DNA molecule, which is the first model, first one from which we are able to know the DNA via the helical structure. The two strands DNA molecule made by and they are intertwined around each other. That concept was given. These parts I will deal later on by detail. But prior to that, this is exactly historical background or historical contribution made by different scientists in respect of the studying the discovery of the DNA molecule or the genetic substance. Okay, these are the contribution made by different scientists. What name is relative? Frederick Mitchell, Altman, Hartley, Watson Crick, Rosalind Franklin, Maurice Wilkins. These were the names associated with the discovery of the DNA molecule. But now let me speak about the structure of the DNA molecule. Okay, chemical study of the DNA molecule was done during the mid of the 19th century, 20th century, exactly from 1950 onward. The chemical investigation in the starting part of the 20th century also. So Levin and other workers, Levin and his co-author discovered the chemical structure of the DNA molecule. But chemically, DNA and RNA both are similar because both are nucleic acids. We call them as nucleic acids. Nucleic acids are always, exactly, what is the chemical nature of nucleic acids? Nucleic acids are biopolymers. They are biological polymers, biopolymers of higher molecular weight, higher molecular weight, with nucleotides as repeating units, nucleotides as repeating units. Okay, this is the definition that you can use or you can note it down in your notebooks if you are studying or if you are staying with this. Okay. Nucleic acids are biopolymers, polymers of biological origin of higher molecular weight with nucleotides as repeating units. I think you have studied about the structure of nylon or polythene, carbohydrates of different types like starch, glycogen, etc. Every of these molecules are polymers. Polymers are always made by similar or different kinds of small units. They are called repeating units. So here, nucleic acids are nucleotides of polymers of nucleotides. So better we can call them as polynucleotide chains. Polynucleotide chains. Nucleic acids are polynucleotide chains. Nucleic acids are polynucleotide chains. Means nucleotide, many numbers of nucleotides join together form chain. Many number of nucleotides join together form chains from what we call nucleic acids. Okay, so exactly therefore, poly means many and nucleotide is one repeating unit. The structure major unit by which nucleic acids are made is nucleotides. Okay, so these nucleotides are, what are nucleotides exactly? What are nucleotides? What are these nucleotides? What are nucleotides? Okay, nucleotides are, again, a reminder, they are trimers of three different types of molecules. Three different types of molecules. Means nucleotides themselves are trimer. Means made by one molecule made by three molecules. The one molecule. Each nucleotide is a trimer made of three molecules. But these three molecules are one kind of nitrogenous base, a nitrogenous base. Secondly, a pentose sugar, and thirdly, a phosphate group. Okay, that means nucleotides are trimers of three different types of molecules. There are three different types of molecules by which a nucleotide is made. One nucleotide contains, that means a nitrogenous base, minimum, a pentose sugar, and a phosphate group. Okay, that means a nucleotide is equal to a nitrogenous base. plus a pentose sugar, a pentose sugar, a pentose sugar plus a phosphate group, a phosphate group, a phosphate group, a phosphate group. A nitrogenous base, a pentose sugar, a phosphate group. Together one nucleotide. But you note here, these nitrogenous bases are their heteropolymers of two different types again. Bases, actually nitrogenous bases present in nucleic acids means in polynucleotide chains, means RNA DNA. So you want note here RNA and DNA, RNA DNA, both are nucleic acids, both are nucleic acids, both are nucleic acids. Means that both are polynucleotides, both are polynucleotide chains, both are polynucleotide chains, both are polynucleotide chains. Polynucleotide chains means both are chains of nucleotides. Each nucleotide in turn is made by one nitrogenous base, one pentose sugar, one phosphate group. Each nucleotide in turn is made by a nitrogenous base, one nitrogenous base, one pentose sugar, one phosphate group. Okay, these nitrogenous bases are again two types present in nucleic acid. There are again two types present in nucleic acids. Present in nucleic acids. I am writing nucleic acid this way. There are two types of nitrogenous bases present in nucleic acid. দুই ধৰণৰ nitrogenous base nucleic acidত পোৱা যায়। ইয়াত nucleic acid বুলি কওঁতে আমি RNA DNA দুইটাকে বুজাইছো। গতিকে কনফিউজনত নাহিবা ইয়ালে। RNA DNA দুইটা nucleic acids আৰু দুইবিধেই হ'ল polynucleotide chains। কিন্তু polynucleotide মানে many nucleotide. One nucleotide is made by nitrogenous base, one pentose sugar, one phosphate group. কিন্তু one nitrogenous base আকৌ দুই ধৰণৰ পোৱা যায়। nucleic acidত মানে RNA DNA আমি দুই ধৰণৰ nucleic acid, দুই ধৰণৰ nitrogenous base পাওঁ। These two types of nitrogenous bases present in nucleic acids are one type we call purine base, one type called purine base, and the other type we call as pyrimidine base. Pyrimidine base. Purine base and pyrimidine base. The purine base and pyrimidine base. These are two types of nitrogenous bases present in each nucleotide of each nucleic acids, DNA and RNA. Each nucleotide is nucleic acid, purine base. A purine base is characterized by, remind one thing again, a purine base is, these are very important information and minute level I am describing here. The minute level one, no doubt you have studied to some extent about the nucleic acids in class 11. But again, this part needs repetition here because this chapter is the fundamental for entire genetics or biotechnology. So, jiman para himan details is chapter pehla amar aru moyu jiman para himan tomar kobole bicharisu. It will require many times, I think several periods. It will require approximately 40 or 45 periods this chapter will require. Okay. So, purine base means a purine base is a heteropolymer of two rings. A purine base is a heteropolymer of two rings, whereas a pyrimidine base is a heteropolymer of one ring. But both are heteropolymers. Two rings heteropolymer, single ring heteropolymer. But they are derivative of benzene exactly, hexacyclic rings they exhibit. So they are benzene derivatives, or we can call benzene derivatives. The purine base, again you remind. Here. This is the finish of a nucleotide. Now let me to continue. We have learned that a nucleotide contains three different types of molecules. There is a nitrogenous base, there is a sugar, pentose sugar, and there is a phosphate group or phosphoric acid. A phosphate group means phosphoric acid. Okay. Every nucleotide is made by three different types of molecules: a nitrogenous base, a pentose sugar, and one phosphate group or phosphoric acid molecule. Okay. These nitrogenous bases are two types, purine and pyrimidines. Purines and pyrimidines are two types of nitrogenous bases. The purine base is characterized by two rings, heteropolymeric two rings, benzene derivatives. Actually, two heteropolymeric rings are present. On the other hand, in pyrimidine, there is only one heteropolymer. Benzene ring is the heteropolymeric ring structure. Heterocyclic benzene ring. Heterocyclic benzene ring also you can use the term. So, structurally, a purine base contains two rings. One is heterocyclic benzene ring associated with one pentacyclic ring. This in skeletal formula for a purine base is this. One purine base is characterized by one heterocyclic benzene ring where these positions are occupied by nitrogen. Here there is a nitrogen. This position occupied by nitrogen. This position is occupied by nitrogen. This position is occupied by nitrogen. But the other positions, other points, other corners are occupied by carbon. Here carbon. Here carbon. Here carbon. And here carbon. And here carbon. This is a purine skeleton. A purine, we can write PU, purine skeleton. Purine skeleton is this molecular purine skeleton. On the other hand, a pyrimidine skeleton is only one heterocyclic benzene ring. Only one heterocyclic benzene ring if pyrimidine base is made. Here, this position is occupied by a nitrogen, and this position is occupied by a nitrogen again, but there are carbons in the remainder corners of the molecule. Carbon here, carbon here, carbon here, carbon here. Okay? So, the purine, this is actually pyrimidine skeleton. Pyrimidine or pyrimidine, PY. Pyrimidine skeleton. Pyrimidine skeleton. Okay? Now, in nucleic acids means combining RNA and DNA both together. Nucleic acids are two types, RNA, DNA. In RNA as well as in DNA, the purine bases are of same type present, and there are two types. Means purine bases are two types, both are present in RNA and DNA. Purine bases are two types, and both are equally present in, if not equally, both are present in RNA, DNA. Maybe equal or unequal, this is not doesn't matter. Molar proportions I am not explaining here, but RNA contains both purines as well as DNA also contains the same purines, whatever RNA possesses. There are two types of purine bases are present in nucleic acids, RNA, DNA. One is called adenine, and the other one is called guanine. Other is called as guanine, adenine, guanine. Two types of purine bases present both in RNA, DNA. Repeatedly, I am repeating the information similarly. Mane RNA, DNA, purine loi কোনো difference আমি নাপাওঁ. Adenine, guanine, iron to থাকে, DNA to থাকে. গতিকে এই মলিকুলটো লৈ পেলাই এই মলিকুলটো অৱশ্যে মই একে কমপ্লিট দিয়া নাই. Adenine is a purine, guanine also is a purine. Means adenine আৰু guanine structure skeletal formula এইটো থাকিব. কিন্তু এতিয়া মন কৰিবা adenine contain here a molecule of amine group by a radical of amine, NH2. Here there is a double bond present. Here is an H, and there is a double bond here, and there is a double bond here. There is an H here. There is a double bond here, and there is an H here. Okay. So adenine is characterized by this bicyclic ring. Adenine is characterized by this bicyclic ring. Caption therefore should be adenine here. Adenine. We can represent this. This is the structure of adenine molecule. Next one is guanine. Is same molecule guanine, also a purine molecule present in both RNA, DNA. But guanine is characterized by, but you remind one thing, adenine, guanine both are purine molecules. But guanine is slightly different than adenine because guanine contains NH2 group in this position. Guanine contains NH2 at this position, but a double bond is absent here. Instead, the carbon contains an oxygen atom here. carbon contain an oxygen here. Okay, but the remainder parts are similar. So this will be now not adenine, this will be now structure of guanine molecule. So note this down. Structure of adenine already I have proposed. Now structure of guanine I have proposed here. Okay, so structure of two molecules, purines and pyrimidines I have given. In comparison to purines and purines, the pyrimidines are three types present in nucleic acids, means DNA and RNA. There are three types of pyrimidines. This is a skeleton of pyrimidine. The skeleton of pyrimidine, RNA, DNA contain three different types of pyrimidines. So PY bases are three types in nucleic acids. This is one is called cytosine, the other one is called thymine, and the last one is called uracil. But you remind one thing here, while there is no difference found between RNA DNA in terms of purine basis, but there are differences found between DNA RNA in terms of the pyrimidine basis. That means cytosine and thymine, these are present in DNA. Cytosine along with uracil present in RNA. That means it will not go about RNA does not possess thymine. Okay. DNA does not possess uracil. So how the bases that present in DNA are purine bases, adenine plus guanine. Bases present in RNA, purine bases, adenine plus guanine both. But pyrimidine bases present in DNA are cytosine plus thymine. In RNA, cytosine plus uracil. But this difference you must note. This difference is purines are pyrimidine. Okay? Here is it.